Hi everyone, I'm Rishik and today I'll be going over the causes, effects, and diagnosis of Alzheimer's disease. Before that, I'll do a quick recap of what Alzheimer's disease is. Alzheimer's is a neurodegenerative disease. It's a form of dementia which is eventually fatal because of its progressive and irreversible degeneration of the brain. Symptoms usually start occurring in the mid to late 60s, though forms of it can appear in the 30-year-old range. In this image, you can see the degeneration of a brain in an advanced stage of Alzheimer's as compared to a healthy brain. A lot of mass is missing from the diseased brain, and the folds and grooves of the healthy brain are mostly missing in the advanced Alzheimer's brain. So, what exactly causes this degeneration to happen in the first place? We aren't exactly sure yet, though it is most likely due to a combination of heredity and genetics, environment, and habits. Head trauma is also linked to a higher risk of Alzheimer's. The first changes in the brain start appearing around a decade before the symptoms of Alzheimer's appear. Fewer neurotransmitters are produced, and the axons and synapses of neurons become damaged and are eventually destroyed in later stages. The two suspected causes of this are neuritic plaques and neurofibrillary tangles. Let's start with neuritic plaques. Neuritic plaques, also known as extracellular senile plaques, are clumps of amyloid beta formed from many incorrectly cut fragments of APP. APP is short for amyloid precursor protein, and it is a surface protein found in neurons. Normally, APP is cut into a form that is soluble, so it dissolves into the fluid in the brain. This form can also bind to the receptors of neurons, preventing them from eroding. However, in people with Alzheimer's, APP is cut incorrectly. This incorrectly formed cannot bind to the receptors of neurons, so those receptors start to erode. This form is also not soluble and clumps up with others outside of the neuron, forming a neuritic plaque. Neuritic plaques also cause neurofibrillary tangles to form. In the image on the left, we see dark spots inside of brain tissue. Those dark spots are amyloid plaques. The drawing on the right shows an example of a beta amyloid plaque outside of a neuron. If another neuron was on the other side of the plaque, a signal from one to the other would likely be blocked by the plaque. Now let's talk about neurofibrillary tangles. All cells have a skeleton inside them to support their structure, just like we do. This support structure inside of a cell is called the cytoskeleton. Tau proteins are integral proteins to the support and structure of the microtubules that make up the cytoskeleton. When a neuritic plaque forms outside of a cell, a reaction is triggered inside of the cell, changing the shape of tau proteins. This new shape no longer supports the cytoskeleton, which falls apart. The tau proteins, no longer connected to the cytoskeleton, clump up inside of the cell, forming neurofibrillary tangles. So let's look at an example of this. In this image, we can see a healthy, diseased, and dying neuron. The healthy neuron is able to support its large branching shape. The diseased neuron is shrinking, and the dying neuron is not able to support its shape at all. In the pop-out images, we see the difference between the microtubules of the healthy and diseased neurons. The healthy microtubules are strong and can allow transport proteins to move along them. However, the microtubules without tau proteins begin to disintegrate, no longer supporting the movement of transport proteins. In this dying neuron, the cytoskeleton has completely disintegrated, which is why it cannot support its previous shape. The death of neurons and the blockage of communication between neurons is what causes the symptoms of Alzheimer's disease. If you want to learn more about these symptoms, go check out the other video on our channel. So, how is Alzheimer's diagnosed? Mental deficits are not clear indicators of Alzheimer's disease, although they are caused by it, because similar deficits to the ones caused by Alzheimer's are also caused by many other conditions, such as Parkinson's disease, strokes, tumors, or even infections. Amyloid beta and tau proteins are current candidates as biomarkers for Alzheimer's. However, there is currently no biomarker to diagnose Alzheimer's in its early stages. Other potential methods of diagnosis are genetic risk profiling, examining cerebrospinal fluid and blood, and neuroimaging. Neuroimaging uses a radioactive chemical marker to bind to neuritic plaques and show their location through a scan. But amyloid plaques are present in the brains of people without Alzheimer's too, so it is not always accurate. Thanks for watching this video. I hope you enjoyed it and learned something about the causes, effects, and diagnosis of Alzheimer's disease.